But Chevrolet.
Oh, sorry.
Hi Jamie, how are you? Let me make you moderator. Hi Z, how are you today? We will start in around 5 minutes or so. Hi Denise, how are you today?
Hey Katerina, feeling a little better. How's everyone else?
Hey Denise, long time no hear. How are you doing, mate?
It's been a rough couple of weeks. Having caught COVID again, but trying to stay in good spirits.
I'm sorry to hear that, man. Hope you feel better soon.
Yeah, I hope you feel better soon. Hi Eric, how are you? Thanks for coming.
Hello. Can you hear me?
Can you
Hey, Eric.
say something again?
Can you hear me?
Yep.
Okay.
Yeah, we can hear you. Thank you. We'll start in around 5 minutes if that's okay.
Sure.
How was your day so far? Everything good?
Pretty good. Nice weather here, finally.
Yeah, right. Took a while.
How did your day finish up, Katerina? Did you get everything done you wanted?
Mostly that I had on my to-do list. Most of it, yeah. Thank you. Actually, Serena gave me more to-do list, which is fine.
Oh, Serena. Does that have anything to do with astrocytes?
Imagine
now
that.
We're here. But it's fine. It's interesting. That's good. It's a good one. Victoria will also be here in a few minutes, she said, and then we can start.
Also, when you get a second, Katerina, I sent you a wee message in the back channel. I'd like to hear what your feedback is on that, because I was wondering if I was going crazy when I was in this other room. Just want to hear your thoughts on that. How you doing, Serena? How's your day been?
Doing good. The day just went by like poof, and here we are.
Yeah, I know what you mean. I've been thinking it's like 5 hours earlier than it is all day.
Well, I'm really looking forward to this talk. It really looks exciting.
Absolutely.
This was a fun time in science when all the experiments seemed to work, and they're sort of few and far between, but it was fun experience.
Oh wow! All the experiments. You don't hear that often.
Oh, and Serena, you really have to listen to the replay of the one that was a few hours ago. That was so good.
I saw the commentary. It really looks interesting.
Yeah.
Hi, Victoria. How are you today?
Hello. Hi. I'm well, thank you. Nice to see everyone. How are you all doing?
Buongiorno, Victoria. How are you?
Great.
Feels like it's been a really long time since we've all been together.
And it's only a couple of days.
It's been days.
It's too long.
I agree.
It's really funny you say that. With me only being in Clubhouse like 6 or 7 weeks, and I somehow feel like I've known you, the group, for like such a long time.
Yes, we're the
So
science
many,
family. That happens.
yeah,
That's really
just so
nice,
much
Jamie.
ground's
Me
been
too.
covered. Yeah, yeah, just so much ground's been covered in such a short period of time. Yeah,
Yeah,
that's
especially
incredible.
astrocytes.
Yeah.
Oh, you know it.
Oh, her ears perked up. Yeah.
I'm kind of hoping to bring back the tardigrade myself.
Oh, and where do we put them all?
Well, they offered to
On the
come
moon.
back, so I think they would be open to come back. It's a while ago they've been here, so we can invite them again.
Was I around for that one? I don't know if I
Was
remember
this
that
the
one.
one where they
That rings a.
quantumly entangled a couple of them?
Yes,
Yes.
exactly.
Qubit.
Ooh.
Jamie, were you? No, I think you weren't here yet.
Was
The first time I met you was when we had the speaker from Glasgow.
Scotland. Yep.
Right.
Glasgow. Yep.
And
Liz
you appeared
Mackenzie.
like
That was fun.
magic. That was unbelievable. Oh, hey, I'm in Scotland too.
So I wonder if we're going to quantumly entangle islet cells.
Well, the thing is with the tardigrades, you can freeze them like this, and then they are still alive. But yeah, with this protocol, Eric, do you think we can do
Well, this
that?
is all, the tardigrade is like the ultimate in cryopreservation. So
Or do the islet cells become entangled as they develop?
it's beyond my physics level.
I want to ask how you quantum entangle something else, but I've got a bad feeling that would actually lead to a whole other room. To just if we started talking about that.
For a Friday, perhaps.
Yeah.
For a
Yeah.
short room on a Friday. Sorry, that's a joke.
Entangled Fridays.
Yeah, should we start?
Let's
I think
do it.
everyone is here, and yeah, let's start. Welcome everyone to the Science Society. Thank you for coming. And today we have our very special guest speaker, Dr. Eric Finger here, and he will talk about this really amazing breakthrough for diabetes. My family has a lot of diabetes, like type 1, so especially interested. And yeah, let me tell you a little bit about Dr. Finger. He is an associate professor in the Department of Surgery at Mayo Transplant Surgery in Minneapolis. He is a transplant surgeon. He did his master's at the Harvard Medical School, Boston, and his residency at the Massachusetts General Hospital in Boston, and his fellowship at the University of California, San Francisco. And he was also a postdoctoral research fellow at the University of California, San Francisco Diabetes Center. And his clinical specialties are kidney, liver, and pancreas transplantation, islet transplantation, and hepatobiliary surgery. Yeah, you covered a lot of specialties and research and complicated medical surgery. So yeah, it's an honor to have you here, to get to know you, and to hear about your research today. But before you start, usually Victoria asks a couple of general questions, if that's okay, and
Sure.
then the stage is yours. Thank you. The stage is yours, Victoria.
Thank you very much, and welcome, Doctor. I'm really glad to welcome you to Science Society, and we really look forward to learning about your research into pancreatic islet transplantation. I looked over the slides, and they're just beautiful and amazing, and so I look forward to hearing you explain it to us and talk about vitrification, etc. It's going to be great. To help introduce you to the listeners, I really appreciate being able to ask a few introductory questions. So the first one is, if you can look back through your life, can you reflect on a moment or an experience that drew you into the sciences? You know, maybe it was a class or a relative, but something that really set a spark that let you know that science was a place for you.
Sure, sure. I apologize in advance. My 4-year-old has just tried to join me, and he's supposed to be in
Oh,
bed.
this is a child-friendly place.
So my sort of interest started. I had an uncle who was a plastic surgeon who did hand surgery, and my cousins and I would go and we would look in his medical textbooks, and he would tell us all the stories about fingers that got cut off and the like, and how they would sew them back on. And that was sort of the beginning of my interest in medicine. And then, but I was an engineer in college and a chemical engineering major. And then it was in a summer research time at Lawrence Livermore Lab in Livermore, California, when I saw how the clinical people addressed sort of more basic questions, but had a really good basis for doing so. You know, they asked the questions not just, what is the tool we're working on, but how can we use that tool to solve some problem? And that's when I sort of saw that combination of science and medicine is really being something that could be quite productive. And that's sort of what began my career, I think.
So perhaps your 4-year-old will find some really fascinating book and get, feel that connection too. And I'm hearing that your interest is in problem solving, and I know that so many of our listeners today are like you, I'm sure, really interested to hear the hope that you'll bring to this rotten disease. And so can you share an overview of the path that led you to your current research?
Sure. It's really sort of an interesting one because it was a side project, if you will. So I'm a transplant surgeon and I do, several different organ transplants and some islet transplantation. And my research had been for many years in transplant tolerance, trying to figure out a way to do transplants without immunosuppression. And we do lots of small animal models for transplant to study various immunosuppression medications and treatments. And someone came to me and said that the mechanical engineers they want to apply for a grant to cryopreserve limbs for people in the military that get their arms blown off or something like that, and they want to apply for a grant to try and cryopreserve an arm or leg. And you do these animal transplant models. Why don't you go talk to them? And so I went to talk to them, and I said, well, this is really interesting. But a leg has bone and nerves and blood vessels and fat and all sorts of different kinds of tissue. That might be a little bit too challenging to start. Why don't we think about something a little simpler, like a kidney, where you have one blood vessel going in and one blood vessel going out, and see if we can't do that. And it's one of those fortunate sort of sidetrack where we applied for a little seed grant, got a little bit of money from the university, and then got a little bit more money from the university, and then we got our first R01 and our second R01 and then our third R01, and then a NSF, multi-million-dollar sort of thing, and it's turned into many people with many different contributing specialties with engineering, medicine, chemical engineering, radiology, all sorts of different people participate. It's really been quite an interdisciplinary research effort.
Yes, I saw the list of names, and it is really extensive. Well, thank you for bringing us up to present, and the way that we do the room. I'm sure you heard in your intro. However, whichever way you're comfortable, if you prefer to deliver your research and then have a Q&A afterward, that's great. And if you prefer to have people asking you as you go along. That's completely up to you. In any case, the mic is yours, and just to know that when you see people flashing their mics, that's a form of applause. So like that. So you know we're with you. So yeah.
Oh, well, before we begin, is there an access issue with the link?
Let me go ahead and check.
Have other
Yeah.
people had a problem accessing the PowerPoint or the
I was
PDF?
able to access it.
Yeah, it should be. I checked again. Oh, let me see.
Thank you, Serena.
Okay.
Welcoming doctor, I just sent out a
I,
request to you.
anyone that is not like our regular, Dr. Shah, can you try to press on the link again?
Perfect, it works now. Thank you so much.
Thank you so much.
Go team.
Okay,
Ready?
take us away.
Sorry.
Ready, go.
Well, really, it'd be great to be informal, and certainly if you have any questions at any point, just jump in, because the slides are a little bit more of a discussion point than specific details, and so I'm happy to talk about things in general terms.
Thank you. Do you know that you're muted?
There we go. Sorry about that.
No problem.
So
Cool.
I probably said that. So it'd be great to just be informal. Certainly jump in at any point and ask any questions that anybody has. And if you the slides are sort of a little bit of a discussion points rather than details that need to be followed or anything like that. So certainly I'm happy to go in any direction people want to go, and we can chat. So to start with, if we go to slide 2 is the sort of the introduction to what's called the Edmonton Protocol. And the Edmonton Protocol was a paper published in New England Journal in the year 2000 by a group from Canada, from Edmonton, by this guy James Shapiro and a bunch of colleagues, where they sort of revolutionized the way we do islet transplantation. And islet transplantation, as you may or may not know, is you take the whole pancreas and you digest it up with collagenase, and then enrich the islet. So those are the the collection of cells that contain the beta cells that secrete insulin. And what they found in that paper was by modifying the immunosuppression in some of the ways that they did the isolation, they were able to transplant 7 patients and get all of them off of immunosuppression, which was great. The problems were severalfold. First off, they had them all off immunosuppression for a year, but almost all of them were back on immunosuppression by the following year, by the second year. The second problem was that you need a fairly large number of islets to get off of insulin. There are about 1 million islets in the pancreas, but the process of isolating them is not very efficient. And so a typical islet yield might be 200 or 300 or 400,000 islets. And we think you probably need more like 700,000 islets to get some off of insulin. And so they were needing to do transplants, 2 or 3 transplants, or even more transplants to get people off of insulin. And so that required several rounds of surgery. It's a minor surgery. In this case, they did by interventional radiology, but we do ours by a small surgical procedure. So 2 or 3 surgeries at least. And then the islets the diabetes wasn't quite durable enough. You get about half the patients come back on insulin by a year, and it really wasn't really durable over a long period of time. And then also people had to be on immunosuppression medication for as long as the graft was working, and that has a range of complications. Just gonna let my wife know.
That's really sweet.
So that was a problem. So you needed multiple procedures, multiple doses of islets. So each dose came from a single donor, and so it was a challenge for many different reasons. And so one of the things that we thought we might be able to do would be to store islets for a period of time and then bank them up and then give 2 or 3 donors worth of islets at one time. We could get more islets in, they'd be more effective. Patients would only have to go through 1 round of immunosuppression induction, and so it'd be less risky. And also it could help with protocols to try and induce tolerance. So if you were to do something to modify the immune system, where you needed to start the patient off and do a treatment for a period of time and then do the transplant in a week or 2 or in a month, it gave you a way to store the islets for a period of time. So that's sort of the basis of why we do it. Also, the other thing to do is, other way to overcome this would be to use stem cell derived islets. That's really been a big current avenue of research to try and figure out a way that you could turn stem cells into islets. And you can grow big vats of them, which is great. The problem is that not all of the batches of islets work. And so you need a period of time to figure out which islets are going to be the good ones to transplant, and the islets deteriorate in culture. And so there was a need to be able to try and cryopreserve islets for that reason, for stem cell derived islets for quality control measures. If we go to slide 3, this is sort of a general approach that we take. So we're talking about cryopreserving islets here, but this is sort of more broadly applicable to a lot of different things. We also try and cryopreserve organs for other types of transplants and tissues for regenerative medicine and things. And you look at different opportunities for preserving tissues. And in transplant, there's a lot of work these days to do what we call normothermic perfusion, which is to pump warm oxygenated blood through an organ. And that might extend the preservation time of an organ for a day or 2, but you still only have a couple of days in order to get the organ in. Islets, on the other hand, you can culture for a few days, but when you go beyond a week or 2, they start to lose their function. And so there really isn't a good long-term culturing or cold preservation method for islets. If we go on to slide 4, and actually I'll just go ahead and skip to slide 5. You mentioned tardigrade earlier. What we can do is we can take some cues from nature to figure out how we might be able to cryopreserve tissues. And nature has discovered lots of different ways to do so. Tardigrade is one example where you can basically dehydrate your body, or the tardigrade body, and remove all of the water, and so that if you don't have any water in the system, then you can't form ice. And ice really is the enemy that we seek to avoid in cryopreservation. For the most part, you either have to avoid ice, as we'll talk about doing, or do what in the bottom left, if you see the wood frog, the Rana sylvatica, that's a species that's adapted to cold weather environments, and it can survive freezing down to -20 centigrade or even lower for short periods of time. And the way that it does it is it modifies the biology of its cells. It changes the composition of the lipids and the cell membranes. It accumulates an antifreeze or urea in the blood, but about 2/3 of the frog can freeze completely, and then when it warms back up several months later, it can come back to life. And so it's adapted another strategy, which is not necessarily avoiding ice, but figuring out a way to manage the ice so that it's in the blood vessels and not in the cells. And so one of the main strategies is either avoiding ice, and the second one is to manage the ice. If we go on to slide 6, this is the traditional way that we cryopreserve things. If you've been in lab and you've tried to cryopreserve cells in a cell line or whatever, what you do is you take those cells in suspension, you mix in some cryoprotectant agent, and that's something to stabilize the cells, usually DMSO, and then slowly cool them at about a degree centigrade per minute. What starts to happen is that ice forms outside of the cells. And as ice forms, whatever solutes are left in the solution surrounding the ice becomes more concentrated. And so the process continues to ice expand, and then the remaining channels of fluid become more and more concentrated with solutes. And what that does is draws water out of the cells. You can see that in C in the middle of the figure. And that when the water leaves the cells to sort of equilibrate the high concentration of solutes outside the cell, it dehydrates the cell and shrinks it. So now the concentration of solutes inside the cell is higher, and that inhibits ice formation inside the cell. And so it eventually will get to a point when you essentially don't form ice in it. And we call this conventional or slow cooling. Ice is formed, but the ice occurs outside of the cells predominantly. And it's tolerated by cells in suspension, but not by cells in aggregates. The problem is the ice is damaging in itself, but also you can form fractures and things like that to destroy the aggregates. It's more of a problem with organs, where we can crack organs, than it is for, say, islets, which is a cluster of a few thousand cells. But it's still an issue that we haven't been able to overcome with cryopreservation or conventional cryopreservation. People have tried for decades to cryopreserve islets because of this. They would like to pool multiple donors and then do the transplant or do the quality controls like I mentioned. And they haven't been successful, and that's partly due to the injury from ice, but also due to the injury from the cryoprotectant agents that people use. And certainly anybody jump in with questions as you if you have them. But so one strategy that we've known about for decades, really about 50 years maybe now, is instead of freezing a sample, it's to vitrify it. And vitrification is basically to turn a liquid into a glass-like state. And now we're looking at slide 7. And what you have is a phase diagram on the left of water, and then sort of a schematic on the right of what happens. And so the details about the schematics are not important, but basically what we know is that as you cool water or any other liquid, when it gets below its freezing point, or its melting point, it's a more appropriate term, you have the tendency to form ice. However, we know that there's a range below the melting point where the fluid still remains liquid. We call that a supercooled state. And we know that if you take a bottle of water, you put it in the freezer, there's a period of time when it won't form ice unless you shake it up. If you shake it up, it'll rapidly form ice. And the reason for that is it's energetically unfavorable to undergo the phase transition from liquid to solid. It requires an activation energy to overcome that. And so at what we call high sub-zero temperatures, so that's a minus 10 degrees centigrade or something like that, water really doesn't want, eventually will form ice, but it'll just take a long time to get there. As you go lower and lower in temperature, the activation energy is reduced, and you finally get to a point where it'll spontaneously freeze. And that happens, somewhere around minus 40 degrees centigrade. However, if you go down in temperature really, really fast, and you can avoid forming ice while you're cooling, eventually you get to a place at around minus 120 degrees centigrade where the viscosity of the liquid becomes too high for the molecules to rearrange and form ice, or to form a crystalline structure. And that's when it transitions into what we call a glassy phase, or an amorphous solid. So essentially what you have to do is you have to go from 0 degrees centigrade to minus 120 degrees centigrade at a rate that's too fast in order to form ice. And if you get down there, you end at a stable state where it's a glass, and so it's a little bit brittle, but you don't have the destruction formation of crystals. And at that low temperature in liquid nitrogen, say, or at a minus 150 degree freezer, things you could store theoretically indefinitely. The problem is, in order to get down to that temperature without forming ice, you have to cool incredibly quickly. For water, that's around 1 million or 10 million degrees centigrade per minute. And that's just too fast to be practically achieved. What you can do, though, is add things into the solution that inhibit ice and lower the rate that you need to cool. That's the rate that you need to cool is called the critical cooling rate. So you have to cool faster than that critical cooling rate in order to get to a vitrified state without forming ice. The opposite is also true. Once you get to a vitrified state, you have to rewarm that liquid, the vitrified state, to above the melting point even faster, about an order of magnitude, 1 to 3 orders of magnitude faster than you had to cool it, in order to avoid ice actually while you're heating. Because now you start at a low temperature and the system really wants to form ice. And so that's the trick, is to add cryoprotectant agents and have fast cooling and then subsequent rewarming to avoid ice transition. And that enters in what we call the vitrified state. And on slide 8 is a picture from a colleague Greg Fahey that showed this in the 1980s, that if you're able to cool a liquid, in this case it's a rat kidney, you can see on the right, you can see through the liquid around it because that's a vitrified kidney. It's encased in glassy fluid rather than the frozen one on the left, which you can see is discolored. And we'd wanted to do that for islets for many decades, knowing that this could happen. The problem was that you had to add too much cryoprotectant agents, too much of those preservation solutions, which themselves were toxic. So either you had to add a ton of the CPA to prevent ice formation, or you had to cool and rewarm really fast. And no one was able to achieve all those things at the same time. In slide 9, they were talking briefly about the development of cryoprotective agents. And this is really just to show that there's a number of different compounds that can stabilize the liquids in these cryoprotective agents. DMSO is the most common one, but ethylene glycol, propylene glycol, all sorts of different mixtures that are penetrating and non-penetrating, meaning some that go across the cell membrane and some that don't. And what we actually end up doing is we end up using them in combinations. If we look at one on the bottom right called M22, that's a compound of agents that includes DMSO and ethylene glycol and a bunch of other agents. And that changed the critical cooling rate from millions of degrees centigrade per minute to 0.1 degree centigrade per minute. So now you get to a point when you can actually enter a tissue, biological tissue, into vitrified state by cooling at reasonable rates and subsequently rewarming it. The problem with those agents, again, is that they are too toxic, and the islets themselves don't like it. They don't respond afterwards. And if we go to slide 10, you'll see what is basically the crux of the problem, is that you need to add more CPA in order to avoid ice, or you need to improve the heating and cooling ability in order to avoid ice. And so you have to get a compromise of the toxicity from the cryoprotectant agent solutions and your ability to avoid ice by cooling fast and rewarming fast. And that's sort of the balance that we need to achieve in all of these different tissues. And on slide 11, we begin what we sought to do for pancreatic islets. And people had tried to vitrify pancreatic islets, and they tried conventional means for cryopreserving them. And what people were able to do is either cryopreserve them with high viability but in very small numbers, or to do it with agents that you couldn't use in humans, or to do it but lose a lot of islets in the process. And no one had really been able to achieve the combination of high viability, clinically approachable, so FDA approvable means, scalable and functional islets in one method. People could do one of those 4 things, but no one was able to really combine them all into a clinical protocol or scalable protocol. And so we sought to try and do that by applying some of our methods. And the first thing we needed to do was figure out how we were going to rewarm them, how we were going to cool them and rewarm them. And we started out by doing droplet vitrification, which was we would use a 3D printer and print, or just like an inkjet printer, and suspend the islets in cryoprotectants and then print them directly into liquid nitrogen. The problem with that was that the liquid nitrogen vaporizes as it gets warmed up locally as that droplet hits it, and it undergoes what's called the Leidenfrost effect, which is it forms a vapor around it, just like when you sprinkle water in a hot pan, it sort of bounces around because it vaporizes. That same thing happens when you print a droplet into liquid nitrogen. It forms a vapor that insulates it and slows down the rate of cooling, and so you don't get fast enough vitrification to form a vitrified state. You can add various CPA compounds. In this case, on the left, you see ethylene glycol and DMSO with some sucrose as well. You see the little droplet on the left, that's a vitrified glassy state, whereas if you just use a salt solution such as PBS, when you print the droplet into liquid nitrogen, it vitrifies, forms ice crystals. So we're able to work around different ways to overcome that Leidenfrost effect. So we couldn't print directly into liquid nitrogen, but we could print in copper dishes that were floating in liquid nitrogen, and we did a pretty good job at forming little droplets that contained islets. But then we had to figure out a way to rewarm them, and we tried all different kinds of ways to do that. So we had all these islets in little BBs, or little marbles, if you will. An example of that is on the right. You can see the islets are the little cloudy things in the sphere. And we tried things like directly dropping that sphere into warm media, and that worked okay. It could heat at about 15,000 degrees centigrade per minute, but it wasn't perfect, and we were seeing not great viability. So we switched over to a different thing called laser rewarming, where around those islets we put a bunch of gold rods that absorb energy, and then we'd shoot a laser at it, and the gold rods would absorb energy and heat from within that droplet, and we could heat really, really fast. The problem was that the irregular surface or the rounded surface of the top of the islet, of top of the droplet, it refracted the laser beam, and so you got uneven heating where you had dark zones and light zones, and so that didn't work because we lost about 20% viability because there was an inhomogeneous heating. So then we switched to what we're talking about now, which is our most recent thing, and we call it the cryomesh. And this is from our paper, basically a summary of the sort of the whole process. And what we do in the top left in figure A is talking about how we get our islets and the islets that we used for this study. And we used multiple different species. We used pig, mouse, human islets, and we also used stem cell-derived islets. So starting from human embryonic stem cells, we matured the cells up to a beta cell, so the insulin-secreting cell cluster. It's an islet-like structure. So we tested all different types of species. We also done them in non-human primates and other species, and it all seems to work. We worked to try and balance that issue of toxicity of the CPA solution, now we're in the middle, versus our ability to heat and cool fast. And so we worked on the technology, and then we sort of developed some methods and used some other people's methods for analyzing them afterwards in terms of the viability and morphology, the metabolic health, how they did in vitro and how they did in vivo as islets. And on the bottom, we sort of summarize the basic methodology. What we do is we take the islets and we put them in a CPA solution. The CPA solution is a very concentrated solution, and so you have to add it in a stepwise fashion. So you increase the concentration up slowly or over a period of time, which we had to optimize. And that's the orange solution around the islets. The islets are little clusters in the bottom of the tube. With that, though, if you try and vitrify that, there's too much liquid around it, and so it slows down the rate of cooling. So what we did was we used something that we developed for cryopreserving Drosophila, which is basically we took those islets that were then loaded with CPA, we put them on a nylon mesh, and then that allows us to sort of blot away the extra cryoprotectant agent solution. And you can see on the bottom left where the islet sort of goes from having a bunch of solution around it to the islet just having a small little residual amount. And so that lowers the thermal mass incredibly. And so you can take that whole thing and then dip it in liquid nitrogen. The islets stick to the mesh. They vitrify in place because the cooling rate is incredibly fast. And then the reverse is also true. You can just take it out of the liquid nitrogen, stick it back in media, and then they rapidly warm back up. And with that, we can achieve really fast cooling and rewarming rates. And from a theoretical examination of what people have done in the literature, we're able to see where we were able to be successful and other people's were not, because we're able to cool faster than the critical cooling rate and warm faster than the critical warming rate to avoid ice. And we think that other people failed in this attempt because they were either too slow or they didn't have enough cryoprotectant agent. So we'll just sort of skip right to slide. Let's see, I can't even see my slide numbers. Slide with the microfluidics. It's slide 15. So one of the challenges first was to figure out how we can get the CPA solutions in and out of the islets because they're fairly highly concentrated. And so we did a combination of modeling for mass transfer and then also empiric measurements about the behavior of the islets, because when you take an islet and you put it in a concentrated solution, it shrinks because it's in a hypertonic solution. But then the cryoprotectant agents diffuse into the cells themselves, and then the cells shrink back towards the original state. And so by modeling the amount of shrinking and swelling, we were able to sort of predict a zone of safety where the islets didn't shrink too much and then didn't swell too much either, when we took the solution out. So we developed a protocol that we could load and unload the islets with the cryoprotectant agents. And then we had to test a number of different agents and try and figure out which had the ability to vitrify the islets without having too much toxicity. And without going into too much of the details, in the bottom center, you see basically our standard assay to look for viability of islets. And so these are pictures of individual islets looking at CPA only, that's the cryoprotectant only, and then VR stands for vitrified and rewarmed. So islets that have been vitrified to a vitrified state and then rewarmed. And we stain with acridine orange and propydium iodide. And so green cells or teal cells are live and red cells, propydium iodide, are dead. And you can basically see that you have a mixture of live and dead cells. And by mixing different compositions of different compounds and ratios and different agents, we found that by using 22% ethylene glycol and 22% DMSO, we're able to find a cryoprotectant that allowed us to vitrify without forming ice and with minimal toxicity. On the bottom right, you see we get toxicity, we get CPA islet viability that's about 90% or so relative to control, which we thought was adequate to move forward. So we used ethylene glycol and DMSO as our cryoprotectant for the rest of the study. If we go to slide 17, we see after vitrification rewarming what the cells look like. And again, in the middle shows the viability assays. We can see we look at mouse, stem cell derived islets, pig islets, and human islets. And you look at the left column in the center, those are the live control, almost all green. In the middle is the vitrified and rewarmed. Those are essentially all green with just a few red cells around the side that are dead. And on the right is the conventional cryopreservation methods that people have used for decades. And you can see a lot of red dead cells showing that we lose a lot of viability. We examine these by light microscopy on the left, electron microscopy on the bottom left, and a number of different assays. And basically what we see is about 90% viable cells after the cryopreservation and rewarming. We can see that there's a mixture of apoptosis going on in the cells and also some necrosis, meaning that there's mechanical death and also some intrinsic cell death through the cellular processes that lead to cell death through a number of different stains, including tunnel stains and annexin stains. If we go to slide 19, we'll see that if we compare the viability, the islet has a number of different cell types. It has beta cells that secrete insulin and alpha cells that secrete other agents, other hormones and things like that. What we found is that all the cells were equally susceptible, equally viable, that is, so that the viability remained 80 to 90%, regardless of which of the cell type. And so we knew that the beta cells were likely to be viable. And if we go to slide 20, we look at the staining with TMRE, which is a stain for mitochondrial health. And on the left, you see the red staining. If you go across, and we'll just look at the mouse islets. The mouse islets in the top left, that's a live control. The red is a stain that shows healthy mitochondria within the islet cells. In the middle is vitrified and rewarmed, and you see also those are quite red and show good fluorescence intensity, suggesting those are live with adequate mitochondria. And on the right row, the right column, you see a very lucent or dark lucency in the center, suggesting that the conventionally cryopreserved islets lose their mitochondrial function, and that became true over time. In slide 21, I show briefly that these cells have normal respiration. They consume oxygen. This is a seahorse assay to look for the cellular mechanisms for ATP recovery. And we can see through all different panels of treatment that the islets consume oxygen for production of ATP, and they have an adequate spare function, and they appear to be very similar to control islets. So on the left, you see the curves of a seahorse assay, which measures oxygen consumption in response to a number of different stimulants. And basically, you can see the top two curves are the control islets and the vitrified and rewarmed ones. The bottom ones are the conventional cryopreserved ones, and dead islets. You can see that there's a significant difference. And so the islets appear to be normal in terms of their metabolic function. We go to slide 22. This is glucose-stimulated insulin release assays. The short of this is if you take the islets after vitrification and rewarming, you put them in low glucose solution and you measure how much insulin is released, and then you move them over to high glucose solution, and see if they increase the amount of insulin, you can see that we get what is called a stimulation index of greater than 2, which means that the islets secrete insulin in response to high glucose in an amount that's the same as fresh control islets. And we see that for mouse on the left, stem cell derived human islets on the right, in the middle, and human islets on the right. So we show that they function in vitro. Finally, we show in slide 23 what happens in transplant models. So this is a mouse transplant model where we take black 6 mice, we make them diabetic, chemically diabetic. And so we start out with a blood sugar, the normal blood sugar of a mouse is around 100. We treat with these agents to kill off their islets, and then so all the mice start with a blood sugar around 400 or 450 that are diabetic, and then we transplant the islets under the kidney capsule. And what we see is if you use conventionally cryopreserved islets, that's sort of the teal color on the upper part. You can see the blood sugar drops a little bit to around 300, but never really fully cures the islets down to 100. On the bottom, you see a series of curves that are overlapping, which is essentially showing that when we transplant these vitrified and rewarmed islets, that they normalize the blood sugar in the diabetic mice within a day or two, and they stay. In this case, we tested for 150 days. The blood sugars remain normal with excellent glycemic control. We can store them for an hour, a week, a day, or in this case, up to 9 months, and they all function really well in vivo. We did another series in slide 24. We tested human islets as well in a xenotransplant model. We didn't try to normalize diabetes, but we showed that these islets secrete insulin in response to glycemic challenge, and that the appearance on the left is called a glucose tolerance test, where we inject glucose, and we see that the blood sugars rise, and then they fall back down to normal values with the same course that the control islets do. Slide 25, we show some immunocytochemistry, fluorescence microscopy, confocal microscopy, showing that the transplanted islets after cryopreservation express insulin and glucagon, and they seem to be, they have the same appearance as the fresh control islets after transplant. Finally, in slide 26, we show the recovery. We were transplanting in small batches. We would preserve 400 to 2,000 islets, and we would get 97% recovery. So we got almost all the islets back, with viability that was 92%. We increased to 2,500 or 10,000 islets per test. The recovery was reduced a little bit to 92%, but the viability remained high. And right now we're working on the next step, which is scaling this up to a higher throughput system. And we think we'll be able to cryopreserve islets in batches of hundreds of thousands of islets. And we think that will finally achieve a sort of a clinically meaningful amount. So this was on slide 29. This was a lot of people involved, both at University of Minnesota, where I'm at Mayo Clinic, where our collaborators that produce the stem cell derived islets are. And I draw particular attention to John Bischoff, who's my close collaborator in the Department of Mechanical Engineering. And our labs have essentially joined in all this cryopreservation research, and it's really been a quite rewarding thing to have the mixture of engineering that he's a specialist in and some of the more biological aspects that I focus on, and it's really been quite productive. And the final slide, in slide 29, is a bit of an advertisement. As I mentioned, we were quite fortunate in our fundraising. We've gotten several R1s, and finally we got an engineering research center grant from NSF. We have multiple institutions now with multiple targets for cryopreservation, and showing around the side at the top of the circle, we see cell therapy, which this is an example of tissues and organs we're working in quite actively. But these cryopreservation technologies that are based on vitrification have broad application in a lot of different things, such as in coral for maintaining biodiversity, in food stocks to cryopreserve shrimp stocks for aquaculture. There's quite a lot of application of these in a diverse range of fields, and this is just one of those many. So with that, I'm happy to answer any questions that people have.
Wow, that's so impressive. It's really wonderful work, and yeah, you said that most worked. So, um, maybe to answer a question that maybe a lot of people have. So, how many of these islets again would you need for a patient to basically have a insulin level that they would need any injections or so of insulin, and how many would you, so you would get that from donors, I guess. How many donors would you then need for one patient, or is one donor enough?
So that's an excellent question. So in the field of islet transplantation, there's a general estimate that you need about 10,000 of what we call islet equivalents. Islet equivalent is essentially an islet that's normalized to a standard size, which is 150 microns. And so you need about 10,000 islet equivalents per kilogram of recipient body mass. So a 70-kilogram person, we would estimate, needs about 700,000 islets. The human pancreas contains, depending on the size of the person, around 1 million islets. The problem is that the isolation efficiency is quite low, and so a typical yield of after an islet isolation might range from 200,000 to 300,000 islets after an isolation. So you would need 2 or 3 donors worth of islets in order to get people off of insulin. Some centers are better than others at isolating islets, and the main goal about this would be that you could isolate islets and start to put them in a bank. Essentially, you'd have a freezer full of islets, and you'd pick which ones would be the best for any one recipient. You could pick islets that are matched for HLA molecules, and assemble enough that you could have to get someone off of insulin with one procedure rather than multiple procedures.
And would you be able, well, let's say you get diabetes, like age-related diabetes or the later onset type 2. W- I don't know if there's much genetic indication, or how precise the gene sequencing data is to predict diabetes, but would it be possible in the future to isolate as a young person a few islets, grow more of them, and have basically your own bank storage of islets?
Sure, that's an excellent question. I think in order to isolate islets, you would need to take out part of the pancreas, and that's quite a big operation to do. So it probably wouldn't be something you would do with that sequence of events. But what you could do, as you all are aware, our ability to isolate or to induce stem cells, iPS stem cells. So taking mature cells, de-differentiate them to a stem cell-like phenotype, and then re-differentiate them into a beta cell, which is the insulin secreting cell, is made significant progress. And so you could imagine where you might take stem cells or even cord blood cells and develop them into islets and freeze them for later use. That's mainly, I think, where it might go if you were going to do an autologous transplant, by regenerating islet cells. But you still need to preserve them in order to make sure that they work, and that's one of the
Yeah, exactly. Yeah, how long can you preserve them? Do you know? Is there a limit?
We've done it for 9 months, and we think that the preservation should be indefinite, as long as you don't gradually lose water, or the liquid nitrogen doesn't disappear, or cosmic radiation eventually will affect the DNA stability. But theoretically, the storage is indefinite.
Oh, that's great. Okay. Yeah, thank you. Please go ahead with your questions.
Really amazing work. I'm curious, in the scale up, what were the problems that you're seeing between 2,500 and 10,000? You mentioned earlier organ fracture, or what are the barriers to scaling at this point?
So one of the really nice things about this is, because the way we preserve these, the islets are put on this mesh, and it's basically a woven material that's, it's nylon essentially with a pore size, it's 36 microns or something. And theoretically, you can scale unlimited. You're not really limited in your X and Y dimension because it's sort of the islets are, they don't stack on top of each other. So you can just make bigger and bigger meshes. The problem is when we started to scale up, we weren't really quite ready to dip the mesh all in the liquid nitrogen all at once. And so we think we were a little bit slow in getting all of the islets down into liquid nitrogen. So it was just a practical matter about how to logistically do it. And right now we have a postdoc working on this now, trying to make different ways you would do it, where you can make sort of stackable meshes and do in a small volume, do lots of different meshes and do lots of islets at once. And so it's more of a mechanical and practical thing rather than a physical limitation.
Oh, I see. You don't have this mountain of insurmountable failures. You're just getting to it at this
Just
point.
getting to it, yeah.
Okay, wonderful. Just one other sort of esoteric side question. On the cryoprotectants, I noticed trehalose was one of them, and I remember was a scientist at the FDA gave a talk and was really interested in trehalose as a cryoprotect. Have you explored the space with your protocol, or do you really just nailed the ethylene glycol DMSO mix?
So we got the ethylene glycol and DMSO to work, and so I think it's working really well. DMSO people are it's discouraged in general because it does penetrate things, but we use it clinically all the time for preserving stem cells. And so when we do stem cell transplants, I don't know if you go through a bone marrow transplant unit, it kind of smells because all the patients have got cells that got with DMSO, and the people smell. I mean, they'll admit it. You walk around and you'll notice it. Trehalose is a really interesting agent because it has really phenomenal properties in terms of being able to vitrify. It's really quite an excellent cryoprotectant, and it's a non-penetrating CPA, means it stays outside of the cells, which would be good to reduce toxicity, except that it doesn't necessarily prevent ice from forming inside the cells. And so you need to do something also that penetrates the cells and gets to the middle of the cell. The other two limitations about trehalose is it's hard to come by these days. It's hard to order it because a lot of people are working on it, and it's a lot more expensive than things like sucrose. And so sucrose also works as a good extracellular cryoprotectant as well, and it's a lot cheaper.
I see. Thank you.
So thank you so much, Doctor. Absolutely fascinating work. And my question from you is about, you just mentioned about the size that you needed. And I know that in part of the paper you mentioned about more than 100,000. And in the same time, we are considering the islet quality control and the function of the single patient unit, as you just mentioned. So my question is about the requirement for the patient, actually recipient preconditioning, because we know that some of the patient, um, they might, have a kind of infusion or they are dealing with the immunotherapy. So I was just wondering, do you have any further information around the HLA matching and those kind of information that you can share with us?
Sure. So this really parallels what goes on in other areas of transplantation, organ transplantation, kidney and heart, liver, and the like. When an organ donor becomes available, it's irregularly available in an unpredictable way, and you don't necessarily know what the HLA type is going to be ahead of time because whoever dies, and so you basically have to make a at the moment decision whether a particular donor is appropriate for a particular recipient. And just in the way that we do a kidney transplant, we get an offer, we look at it and we say, is this a good enough match for this recipient? If it is, we bring them right in, the organ comes, we do the transplant right away. If we could cryopreserve kidneys, or if we could cryopreserve islets, it would really change the organ allocation, both in policy and practice, because now you could pick that HLA matched donor to better match the donor and recipient. you could better prepare the patient. So, rather than them getting a call in the middle of the night saying come in the morning for a transplant, you could say, okay, we're going to do a transplant, July 5th, and make sure that they're in good shape for that transplant and reschedule if needed. And it changes the procedure from a nighttime one when I'm often operating to one in the middle of the day, which is an advantage for me as a personal note.
And does the changing in size, because we just talk about the cooling and rewarming again, so I know that it has an impact on a mesh size and the shape and geometrical issue maybe. I was just wondering, does it related to the quality of quality control of the islet cells?
So part of the reason is to do, quality control as a normal practice for islets. These days, when we isolate islets, we only have a short amount of time to do our quality control assessments. We can do some in vitro assays, but we can't do in vivo assays. And many of the assays take days or even weeks to get results. And so it's not practical for the fresh batch of islets to do quality control in terms of function. You can do quality control in terms of purity and things like that, but you can't do functional assays, or at least not really good ones in a short period of time. And this lets you do that. It also lets you do. We're also going to have some quality control issues for us to deal with as well, because the islets come in direct contact with liquid nitrogen, and it's you can make sterile liquid nitrogen pretty easily by using ultraviolet radiation or radiation sources, but to remove endotoxin and things like that, it's kind of harder to do. And so we do have a regulatory some regulatory challenges that we need to overcome for our process as well.
Thank you so much.
Oh, I never thought about that problem. That the liquid nitrogen has to be sterile too.
So this is an interesting thing in it's more of an issue in say embryo preservation. So assisted reproduction and things like that. People banking embryos and in vitro fertilization. One of the ways that people cryopreserve those is by vitrification, but they're smaller clusters of cells and they're much less sensitive to toxicity. And those, the original protocols, is they're directly immersed in liquid nitrogen. So this has been a discussion that's gone on for decades, really, to say how do you keep them sterile, but also how do you prevent things from transferring from one batch of embryos to the next, because you could potentially transmit viruses if you just stored them in an open tank. And so there's some barrier things that need to be overcome as well.
Oh, so they are all in one big tank?
So the issue becomes once you, for embryos, for IVF, you can put them in tubes, but often the tubes are not completely sealed. And so there's a concern in liquid nitrogen tanks that they're not completely sterile. And there is a possibility that you can transmit things between tubes in a liquid nitrogen tank that you see or you may have in the laboratory. And so there's a lot that goes on to try and minimize that risk or avoid the risk of cross-contamination. But this is beyond my ability. But people's careers are based on these whole logistics and quality control measurements of cryopreservation in general for stem cell therapy as well.
Interesting. Thank you. I think Mona had a question, and then Steve and Jake joined us. I wanted to check with you how much time you have left, because I know we have in 3 minutes, like the hour is up. I just wanted to ask you if you have time for one or two
Sure,
questions.
whatever people want, or if people want to get on with the evening, it's really
Okay.
my kids are quiet now, so apparently they went to bed.
Thank you. Okay, go ahead, Mona.
Okay. Yeah. Hi, everyone. This is a very interesting topic. So I want to take it kind of a different direction. I wanted to ask about islet transfer and annular pancreas. Is that a viable option to help islet transfers, or so that there can be like a model to see, like how you guys were talking about how nitrogen is not sterile, like those cases, can that be like an organ to test islet transfers in patients who have annular pancreas?
So annular pancreas, for other people that might not know, is the during the embryologic development of the pancreas, there's sort of two different segments. There's the ventral and the dorsal side of it, and they fuse in a normal pancreas, but sometimes they sort of wrap around in sort of a loop shape where there's sort of two components to the pancreas, a front side and a back side, uh, if you will. And that can lead sometimes to people to have some difficulty with the pancreatic ducts that drain the pancreatic juices into the intestine, and can lead to pancreatitis and other challenges that some people have. And though this is not a very common thing, we at the University of Minnesota do a lot of what we call total pancreatectomy and auto islet transplants. These are people that have pancreatitis, chronic pancreatitis, so chronic bouts of inflammation of the pancreas, leading to chronic pain and the need for opiate analgesia. And what we can do for people, it's sort of an end stage treatment option, is we can take out their pancreas. It's a big surgery, but we can do it. Isolate the islets from their own pancreas and transplant them back into the recipient. The problem with that is that sometimes we get more islets than we can transplant all at once. And so we do different things with them. We try and put them in various places in the abdomen. But I think most of those die off. And then many of the patients that get this procedure will need to have some insulin afterwards. And so this is a potential use of this technology where you could cryopreserve a patient's own islets after they've had their pancreas removed and give them back in sort of smaller doses that might be better tolerated over time and have less of a likelihood that they would require insulin down the road. So that's one example of a use case for this.
Okay, yeah, I was always curious about that because it seems like it it can cause those problems, but it can also remain semi-silent, and in some rare cases, like one part of the pancreas can not function properly, and the other one is kind of like protective in some manner of preventing people from getting diabetes in rare cases.
Yeah, no, it's excellent. You know, and you might see how you might do something if someone needed a part of their pancreas removed because part of it was causing trouble, but the rest wasn't. So rather than getting rid of that part of the pancreas, we could isolate islets and stick them in a freezer for that time in the future when they may or may, you know, if they potentially need a islet transplant in the future if they become diabetic.
Yeah, that's super interesting. Thank you.
Sure.
I believe I wanted to check with Ryan and Dennis. You didn't get, and Victoria didn't get to ask a question. But if you don't have one, we'll move on to Steve and Jake.
No questions right now. Just really appreciate you coming out to speak, and thank you for setting this up, Katerina. I'm learning a lot.
Thank you for coming.
Same. Thank you for the author, Katarina. Yeah, this has been fantastic, and I appreciate the clarity that you're delivering this talk with, and I'm happy to pass on.
Well, thank you. If you guys have questions anytime in the future, you know, this has really been a fun time. Like I said, it was this is a side project off of a side project, really, and it's been quite rewarding. And I think that we'll be in clinical trials with this in a couple of years, which is pretty quick for something like this.
Yeah, it is like really quick and fantastic that you say that, you know, that it worked so well, and it's amazing. So congratulations again. Steve and Jake, do you have a last question to ask?
Hi. So no real question. I was late to the room, but listening in, I had to come up. We run a stem cell room every week on Clubhouse, and have a couple of friends. A friend who was shot 26 times and who has had extensive stem cell treatment over the last 13 years, and a soccer player who had his career saved with stem cells after being introduced by this other guy. And so the two of them have been running this room, and we have two doctors there. So, and I was just, excited to hear what you're having to say, Eric, and congratulations, and look forward to hearing more. But I mean, I was also wondering if you'd like to come and join our room sometime to talk. Um,
Sure.
uh,
My work is not too much in stem cells. I use the product of the stem cells. I use islets that are developed from stem cells, and then I also do another project to, as we mentioned in this, that one of the other downsides of islet transplants is you need immunosuppression. And one way to overcome the need for immunosuppression is to combine a transplant like this with a stem cell transplant, like a bone marrow transplant, essentially. And I run some clinical trials here at the university where we're doing a combination of kidney transplant and stem cell transplant with the idea of trying to wean patients off of immunosuppression.
Yeah, I actually right now work for 3D bioprinting of 3D organs or tissues from stem cells. Maybe we should talk one day. Like I used to be just a neuroscience, but now I actually work. I was talking actually with Ryan in the background to maybe print some islets one day.
Sure.
But yeah, we can talk. We can talk one day maybe about it. We did the we are having the first clinical trials with these 3D printed products. So it went faster than we expected that we went into clinical trials. So that's the good news. But it will take us maybe a while to expand to this type of cells. But yeah, Jake, you waited for a long time to raise your to come up here, and Brian, please ask your question.
Oh, yes. Sorry about that. Yeah, I've unfortunately I wasn't able to follow along on the uh, uh, presentation. I've been actually shopping and I'm in the car, and I just wanted to thank Eric. Thank you for your just your passion, your heart. This is well beyond my outside my lane. But I come from a family that unfortunately just had a history of diabetes, and I feel blessed and fortunate to uh, have my great grandparents in my life for quite a long while. But I did see uh, the effects of diabetes take my great grandfather, then my grandfather, and how it's just been assaulting my dad since I was a kid, and it's given me deja vu of when my great grandfather, uh, unfortunately, his health started to decline, and the amputations came, and can't miss insulin. I don't know, can't miss taking his shots and monitoring, his glucose levels, and I'm just thankful for uh, physicians, researchers, and scientists who are really pushing the boundaries for a lot of different ailments that affect all of us in some capacity. And thank God I don't have it. That's one thing that my dad and mom have made sure to impress upon me. But just, I've been listening this whole time, and no question, I just wanted, if you haven't heard it before, I appreciate you wholeheartedly, and just simply thank you. Uh, thank you.
Thank you for sharing the family. Diabetes is a tough one, and the vast, actually, I'd say probably most of my patients, the largest percentage of my patients have diabetes. Because I'm one of my main focuses is pancreas transplantation these days, and it is a challenge. And we do have treatments. The problem is, what is the risk of the treatment? And the good news is that things are changing. People are doing a lot better now than they were doing even a few years ago with even closed loop pumps and glucometers and things like that. But there, we still have a way to go.
Yeah, well, that was a beautiful ending, I think, to this room. I think we can all agree on that. We all thank you for taking the time here today to explain this to us and answering questions, but also for your work. Yeah, and please come back anytime with updates or other projects. You gave such a amazing talk to explain this to us in a way that we could all understand it. So yeah, if whenever you have time again, please come again, and we wish you all the best and that the grants keep coming for all the projects.
It's always better. Well, thank you very much for the invitation, and hopefully after the summer, I think we're gonna have a new story to tell.
Oh, perfect. Yeah, in the fall. That's yeah, in the summer I won't be around much either, so not sure how many rooms we'll have, but yeah, in the fall we'll restart again. So that's perfect, actually. Okay, thank you everyone for coming, and special thanks to you, Eric. And yeah, we'll have again a room on Friday, actually, this week, and on Saturday. If you would like to come, on Friday we have Dr. Singh here talking about artificial photosynthesis systems for CO2 capture. It will be a really interesting room and relevant for climate change. And then on Saturday at 9 p.m. we'll have Dr. Shushani. He's a theoretical physicist at Waterloo in Canada. I'm so sorry, I'm so tired. And he will talk about his theoretical physics work, faster than light travel and time travel. So yeah, please join us again, follow the club, and thank you so much, Eric, for taking the time to come here, and bye everyone.
Can I just say something before you go?
Oh yeah, sure.
Eric, I just want you to know I did send you a message. There's an airplane little symbol on the right. I know you're new to Clubhouse, but if you don't follow me, it's hard to see messages. So if you go to your message system, you'll see a yellow request icon button. So that's where you'll see messages from people who you aren't following yet. So it's a little difficult to see that. But so sorry to hold the room up closing, but thanks.
Okay, sure. Great. Okay, bye everyone. We'll close the room in 3, 2,
Thanks,
1.
everyone.
Thank you.
Thank
Thank you so much,
you. Good
Eric.
night. Thank you, Eric.